Well, good morning, everybody, and welcome to the continuation of the 2015 edition of the Royal Tyrrell Museum Speaker Series. Today, the Royal Tyrrell Museum and its cooperating society are proud to present Dr. Alexander Dutchak. Alex is an instructor in the Department of Earth Sciences at the University of Calgary. He's originally from Edmonton, and he pursued his undergraduate degree in biological sciences at the University of Alberta, where he studied the middle ear structure of a late Cretaceous snake from South America for a senior undergrad thesis. Alex decided to stay at the University of Alberta to pursue a Master of Science degree, during which he revised the systematics of the aquatic lizard Aegialosaurus and of basal mosasauroids more generally. Subsequently, Alex came to his senses and moved away from aquatic reptiles and moved to the United States to pursue his PhD at the University of Colorado Boulder under the supervision of Dr. Jalen Eberly, so a fellow Canadian paleontologist, where he studied the mammalian faunal change that took place during the early Eocene climatic optimum, looking at specimens from basins in Colorado and Utah. After obtaining his PhD, Alex came back to Canada and held lecturer positions in both the Department of Earth and Atmospheric Sciences at the University of Alberta, and then in the Department of Earth Science at Mount Royal University before moving to his current position at the University of Calgary in 2014. Alex's research interests focus principally on patterns of mammalian faunal variation associated with climate change, immigration and emigration corridors between sedimentary basins, and also latitudinal diversity gradients throughout the Paleogene. I'd like to add in a personal note. Alex might be interested to learn, or he might be mortified to learn, that he and I have been colleagues and friends now for over 15 years. Our graduate programs overlapped significantly at the University of Alberta. I TA'd Alex in several of his courses, and in later years he and I actually ended up teaching those same courses together. So I don't know if that says more about the speed at which Alex went through his program or the more tortoise-like approach I took to my program. But either way, I'm delighted to be able to introduce Alex today, and he will be discussing not what is written on my cheat sheet, but actually a talk called The Warm Fuzzies, Mammals and Climate Change in the Eocene of North America. So if you could all please join me in welcoming Dr. Alex Dutchak.
Well, thank you, Craig. It is a real pleasure to be here. I spend most of my time lecturing to undergrad classes, and so it's a real pleasure to be able to step back into paleontology and talk about some of these things with you today. If you came to hear a talk about the Cypress Hills mammals, that's not going to happen today. When I went and looked at climate change and periods of mammalian faunal change, we decided to hold off on the Cypress Hills stuff till next year after we've had a couple of good sessions in the field. So we have a little more data to show you, a little more fun to talk about. So the Cypress Hills fauna deals with cold weather, cold climate or cooler climate. Today we're talking about hot stuff. We're going to be talking about warm intervals in climate. So basically any discussion of Paleogene or Cenozoic climate comes back to this image. And this is a pretty famous image from 2001, a paper published by Jim Zakos. I show this to my classes and I always go off on it because this is, quite frankly, this is an amazing image. This is everything we know about the last 70 million years all in one place. So that's pretty cool. If you want to talk about what's going on climate-wise, you have this beautiful curve over here, which we'll talk about in a couple of minutes. There's your time scale down here, down to 70 million years. There's an estimate of ocean temperature here, which may or may not be super useful. We can look at tectonic events, what's going on, when are various oceans opening. You want to know when the ice sheets start up. Well, we put that on there as well. We're paleontologists, or we're interested in paleontology, so we can talk a little bit about the biotic events going on here. We get mammals dispersing. Mammals have been around a lot longer than that, but they're starting to really take over when we get into the Eocene, although obviously Craig and others study things a little earlier. They're around. Or we can look at the carbon curve down here as well. Now, that's a lot of information, and what's really neat about this is because it's such a big picture, and the summary here, this is information that's been compiled by hundreds, if not thousands of scientists. There's careers worth of work here that you can flip through in a page, which is a little scary, but also it's incredibly useful. So what we're actually going to pay attention to here today, This is the big one we're after. This is the climate curve. What we need to know from here is that the further right you go, the warmer it gets. The further left you go, the cooler it gets. Okay, so technically right now, we're in a relatively cool climate. Now, climate change is those are buzzwords. So climate change is coming on, and certainly things are getting warmer. And so if you wanted to look at, well, what's going to happen, if you wanted to see what happens to populations when things do get warm, well, let's go look at some areas when that happens. We have examples of that in the rock record. So today we're going to be focusing mostly on the PETM, the Paleocene-Eocene thermal maximum, which is this tiny little spike here, or the early Eocene climatic optimum. And that's a little bit longer. Those are the warmest intervals in the last 70 million years. And we have really, really good records for them in North America. However, as we're talking about this curve, we should say, okay, well, you've got this beautiful curve. What does it actually mean? Where does it come from? So here's the same curve. This is our oxygen curve. And there's a timeline down here. So we just turned everything on the side. What we're really interested in here is all these little crosses. That's individual data points. That's a bullet that someone has run through a mass spectrometer to actually get data. And so this is not all from one borehole. What you're looking at here is a compilation of dozens and dozens of studies. Okay? So when Zachos built this curve in 2001, he took everybody else's data and put it back together. And so this data here, where we're really interested in up top, that's the Paleocene-Eocene boundary. That was actually first published by Kennett and Stott in 1990. So this is 25-year-old data. This is not brand new stuff. But we should point out that this is marine data. Okay, so this is coming from deep ocean. It's coming from forams. These are benthic foraminifera. So these things exist at the water sediment interface at the bottom of the ocean. And they build these nice calcite skeletons, calcium carbonate. And so we can take those and add math. And we can back calculate what the ratios of oxygen 16 to 18 were in the deep ocean. And that's what we're actually tracking through here, is the variation in oxygen 16 to oxygen 18. And then we can use that to interpret what's going on with climate at the ocean-atmosphere interface several thousand feet above. Okay, so this climate curve that you get to see every when I'm going to keep coming back to, and you're going to get sick of looking at it. When we see it, remember that's marine data. But what I'm here to talk to you about today is terrestrial continental data. And we have to be able to tie those together. So we're interested down here, the Paleocene-Eocene thermal maximum. And the way we can tie these things together is basically stratigraphy. Now, I spend lots of time talking about stratigraphy to my students. I'm going to assume a small bit of knowledge here that we know that stratigraphy exists. But one of the ways we can do this is paleomagnetic stratigraphy. We call it paleomag. And so what happens is the Earth is basically a big bar magnet. We can run with that. And what that means is that when certain kinds of rocks form, the minerals orient themselves in along that magnetic field line. This would not be useful except for the fact that the Earth's pole switches every once in a while. And so you end up with this barcode of we call it normal and reverse. It looks like this through time. Okay? And we can match that up with sediments deposited at various intervals, add that to radiometric dates, which actually give us numbers, and all of a sudden we have some sort of stratigraphic column that we can start tying this wonderful data to. Okay? One thing we should note in this previous slide here is that the Paleocene-Eocene thermal maximum, the boundary here, according to Kennett and Stott, is around about 57 million years ago. Okay? 57 and a half or thereabouts is where they think the Paleocene ended and the Eocene started. If we come forward and we look here, the Paleocene-Eocene boundary is marked by Zachos at about 54 and a bit, closer to 55 million years. What's happened there is that post-Kennett and Stott in 1990, in 1995 they revised the paleomagnetic time bars, the time scale basically, and they said, oh, we're 2 million years off. And so everything gets shifted. It's a case of science correcting itself. So when we talk about the Paleocene-Eocene boundary now, we talk about 55.5 or 55.8 or 55.6, depending upon who you want to argue with. It's somewhere in that realm, and most people are pretty comfortable with that. Okay, so what that allows us to do is build these wonderful complex stratigraphic columns. So we have paleomag here with our crons, positives and reverse. Okay? We have our radiometric dates here, which we haven't found all of, but we have enough to put a nice bracket on it. And then in between, we've got forams, calcareous nanoplankta, then we've got mammal ages. That's all biostratigraphy. We're using the fossils to tell time. Okay? The example here is from the Green River Basin. This is Mike Smith and his friends. They've done a really nice job of putting together a whole bunch of different basins down in the US and tying them all together. And so this is an example of the power of stratigraphy. If you don't have paleomag, that's okay, as long as you have biostrat and maybe some radiometric dates. Or if you're missing radiometric dates, as long as you have biostrat and maybe some paleomag to back it up, you can still figure out where you are in the time series, and that's the really important part. So we have very robust stratigraphic methods that allow us to constrain the age of the sediments and therefore the fossils that we're going to be talking about. Okay, I realize there's a lot of dots showing up on the screen right now. We will move on to fossils before long, I promise. So here's an example of what you can do with carbon data. This is from Magincola in 2004, and what they're showing here is the Paleocene-Eocene thermal maximum. This is a sharp spike in temperature that lasted 150, 200,000 years, depending upon who you want to ask, and it's associated with a carbon spike. Some would say it's caused by a carbon spike. We'll go with associated. So what you're seeing is your carbon values are jumping here about 6 per mil to the left. This is a terrestrial section found in from the Bighorn Basin. And then we've got it matched up with Belgium and Egypt as well, in addition to Kennett and Stott's data down here from the Atlantic. Okay? So what we can see here is that, okay, great, we've got a carbon excursion. That's fantastic. I'm not really terribly excited about this. What we have is a 1 or 2 per mil jump in the ocean and a 6 per mil jump on land in almost every case. And the question, why does it get amplified on land? The answer is basically we don't know. There's a number of people that have been looking at this, and we're not quite sure what's causing the amplification, but it exists for some reason. The neat thing is that up here, you've got, what's that, about 40 meters of sediment representing that excursion of 200,000 years. If you go to Belgium, you've got about 10 meters of sediment. If you go to Egypt, you've got about 2. So that's just a change in depositional rates. The carbon excursion is still there. It's of the same magnitude, but because depositional rates are different, you're getting different representation in the sediments. If you want to do really high resolution biostratigraphy, this would be the place you'd want to go because you have that lots and lots of sediment. So you're going to get lots and lots of fossils, hopefully, to represent that change through time. Now, if you want to go and do this, the graphs look fantastic and the data is wonderful. Basically, what you do is you go dig a lot of holes. This is a picture of me digging a hole. I spent a lot of time during my PhD climbing up various bluffs, digging holes every meter or so to get sediment from which you can extract various carbon data points to try and do this kind of chemostratigraphy. Sometimes it works, sometimes it doesn't. Chemistry is the geochem tends to be messy. Okay, so back to our big picture. Let's take a look at the Paleocene-Eocene thermal maximum and see if we can tie in some fossils and some projections. So here's roughly what the North America might have looked like at the beginning of the Eocene, late Paleocene, early Eocene. Some major differences. Well, Hudson Bay doesn't exist, so that's kind of something. We're still attached to Greenland. That's pretty important. All the Arctic islands, all attached up in northern North America. So the Arctic islands aren't really there. They're not islands per se. The other big thing over here is that the Bering Strait is really, really narrow, if present at all. Okay? Certainly the Atlantic Ocean up the northern Atlantic up in here, it's pretty narrow. And so what that means is the Arctic Ocean is pretty isolated. The salinity in the Arctic Ocean is probably quite high at this point. Okay? Now, that's going to become important when we start talking about dispersal of organisms and start talking about where the animals that are coming to North America are coming from. But the area we're going to be focusing today is down here in the U.S. montane region, the Rocky Mountain region. We call them the intermontane basins. And if we zoom in there, this is straight off of Google Maps today, we see a whole bunch of holes in the ground and a bunch of mountains where they're looking in all sorts of weird directions. So here's the Uinta Mountain Range, the only real east-west mountain range in North America. That's kind of neat. And there's a whole bunch of basins tied in there where we're collecting sediment. There's the Bighorn Basin up top, which is where we're going to start, because it has a fantastic section that represents or captures the Paleocene-Eocene thermal maximum. Right below it is the Wind River Basin, which we'll talk about in a little bit. Then we have the Green River Basin, which is actually a series of sub-basins, which captures an immense amount of time, and there's a beautiful story associated with lakes that we're just not going to talk about today. But the good thing about all of these basins is they're low spots that are collecting sediment, and therefore we're getting a fair number of fossils. The Green River Basin is famous for a lot of things. Its fossils are one of them. It's also a big oil and gas reservoir. So here's an example of the kind of Lagerstätten you can get. There's a nice little leaf here to give you an idea of some really fantastic preservation. We get very good paleoflora from the parts of the Green River. And then this is one of my favorite fossils ever. We have a little horse up here surrounded by fish. I always give this to my students when we talk about taphonomy. I say, is it those walking fish, or is that a swimming horse? And the answer is it's probably just a floating horse, but until it sank anyways. And so we get very good fossils. There's no rock shop in North America you can walk into and not find a Green River fish. They're all over the place, really good preservation. Another part of the Green River lake system are the Piceance and the Uinta basins, the Piceance Creek and Uinta basins. And so these are the basins we're going to be walking through today, looking at various faunas in various places and talking about different ages. But we want to start with the Bighorn Basin because we're going to go to the Paleocene-Eocene thermal maximum first. Okay, so sharp, rapid global warming. So MAT stands for mean annual temperature. During the Paleocene-Eocene thermal maximum, mean annual temperature jumps quickly, geologically speaking. In the space of 10, 20,000 years, you have a very, very rapid increase in temperature. And then afterwards, you have about 100,000 years where things get back to normal, or basically where they were before. So you're looking at a 200,000, 150,000 year interval where things got hot quickly. Okay? Mean annual precipitation during the Paleocene-Eocene thermal maximum is a subject of a little bit of debate. But generally, during the increased warming period, the 10 to 20,000 years, it's thought that the precipitation dropped significantly by as much as 40%. And then it went back to its 1.5 meters or so a year afterwards. So you're still getting seasonal change. You're getting wet seasons and dry seasons, as evidenced by the presence of various kinds of paleosols, fossil soils. So here's a picture of some people on Polecat Bench. This is an expedition. Just kind of get people out there. As you can tell, this is not a full-grown scientist yet. But so here we are on Polecat Bench with some nice colorful beds. Usually when you see these nice purple beds, what you're talking about is a fossil soil that was subaerially exposed to air. And so it had time to oxidize the minerals within. So periods of dark bands represent good soil formation. Periods of the lighter bands here, poor drainage probably, rapid deposition and poor drainage. That's a generalization, but you're going to be seeing an awful lot of color as we go through these slides. As I drove down into the Drumheller today, I thought, well, those are really nice bedding plains, but they're missing that really pretty red color that I'm used to from the basins down south. So what actually happened during the Paleocene-Eocene thermal maximum at Polecat Bench and elsewhere in the Bighorn Basin? They have a fantastic rock record, a fantastic fossil record. There's thousands, tens of thousands, lots and lots of specimens of mammals. And most of them are going to look something like this. This one's actually from the Piceance Creek Basin, but it gives the general idea. We consider a specimen to be a tooth. If you find a whole skeleton, that's fantastic, but as a mammal paleontologist, I'm really only excited about the teeth to start with. The functional morphologists like the long bones. I'm all about the teeth. So this is from Gingrich. Phil Gingrich has done a whole lot of work, and he's pioneered a bunch of this stuff along with Ken Rose. So the University of Michigan and Johns Hopkins and a number of other universities spend a lot of time in the Bighorn Basin. So Phil Gingrich has this nice stack of specimens. So here we have a whole bunch of specimens. This is ectotion, and you'll notice this is body size. So what we're actually looking at is the size of the teeth, the length and the width of the tooth here on the bottom row, moving up through stratigraphy. And what we can see is that it's generally pretty stable until you hit the Paleocene-Eocene boundary, this WA0 boundary, this WA0 section here. That is the Paleocene-Eocene boundary, the Paleocene-Eocene thermal maximum, and things get really small. And so what you're looking at is dwarfing, possibly, assuming this is all one population. That's an assumption you have to make to start talking about this. And when we see dwarfing like this associated with changes in temperature, cooling, or warming in this case, this may be an episode of what we call Bergmann's rule. And Bergmann's rule basically states that organisms of the same taxon will be smaller when the temperature gets higher. Bergmann's rule is the worst kind of rule ever. It's a biological rule, which means that about 55% of the animals actually obey it. It's a rule in only the loosest term. It doesn't apply to mammals that are less than a kilogram or less than half a kilogram in size. Once things get that small, they can usually go and hide somewhere. They can burrow, or they find other ways to get around the climate. Mustelids, the weasel family, actually go backwards. As things get warmer, they get bigger because they're weird. So Bergmann's rule is a generalization, but certainly it does appear in the fossil record that this does play with our idea of a very rapid warming. So there's some evidence that the fauna is changing. Here we have Heracitherium. Possibly, it's one of our early, you can call it Heracitherium, you can get in an argument, call it Eohippus because it's a better name, whichever. It's one of the early horses, one of the early perissodactyls. And you can see that it also, at the WA0 boundary here, it is very, very small, and then it increases in size, and actually increases significantly in size. These are all called different species, Grangeri, Pernix, and Amylor. That's because they look different. They are different sizes, and when we're in paleontology, sometimes size does matter, and you call them different things because of that. Okay? What we might notice, though, is that unlike Ectosian in the previous slide, there's nothing down here in the Paleocene. Heracitherium just kind of jumps in and says, hey, I'm here at the beginning of the Eocene. There's a good reason for that. There's a number of organisms that do this. Right here in the WA0 boundary, we have a whole bunch of organisms that start to show up. So here's a whole bunch of organisms. Now we have stratigraphy on the bottom on the x-axis here, moving from the Clark-Forkian up into the Wasatchian. So Paleocene down here, Eocene over here. And what we can see is that when we get to the start of the Eocene, a whole bunch of things just show up. There's an increase in species richness, or in this case, generic richness. Okay? You can see that down here. Here's our generic richness curve. It's pretty average, and then you hit the Paleocene-Eocene boundary, and all of a sudden we jump way up into here. Okay? So diversity is increased. That's the WA0 boundary. What's actually happened at the Paleocene-Eocene boundary is we've had an invasion. A lot of those new organisms are actually coming over from Asia. Our first occurrence of artiodactyls, the even-toed ungulates, the perissodactyls, the odd-toed ungulates, so the horses, the rhinos, tapirs, and the primates. It's thought that they come over from Asia. And what's made that possible is that as temperature increases, the organisms that are used to the nice warm temperatures down here in the subtropics are able to migrate north. And eventually they're able to migrate far enough north that they can use Siberia to get across to Alaska. So you're looking at a change in the biogeographic distribution of all of these organisms dictated by climate. Okay. One other thing we should mention about biogeography down here is Meniscotherium. Meniscotherium is this cool condylarth perissodactyl, depending upon who you want to ask. It's this cool organism we find, and in the Bighorn Basin, Meniscotherium is an index taxon for the WA0 interval. What that means is the interval of the Paleocene-Eocene thermal maximum, the interval of this really hot point, that is the only time you find Meniscotherium in the basin. So if you are wandering around and you stumble across a Meniscotherium fossil, you can say, ah, I'm in the WA0 zone. You know exactly where you are stratigraphically. That's great. It's a good index fossil for that reason. Now, if you go down to the Uinta or the Piceance Creek Basin, you find Meniscotherium there much, much earlier than WA0 and significantly after WA0. Meniscotherium's there before, and it's there after. It's also there in between, actually. What's actually happening? It looks like Meniscotherium, as it's getting warmer, to the peak of the warming, that's as far north as Meniscotherium made it. Then it started to cool off again. It ran back south for the equator. And then as it warmed up again afterwards, it made it back into the Bighorn Basin a couple million years later when temperatures had risen again. So Meniscotherium appears to be a nice little paleothermometer, if you will. It's tracking really warm temperatures with its distribution. So here's a picture of Meniscotherium. Here's a picture of, this is the Piceance Creek Basin. Here's my tent. So basically this is us wandering around in the rocks looking for more fossils. And being a more southern of the montane basins, we expect to have different biogeographic distributions, and that's exactly what we see across that boundary. Okay, so that's, we've talked ever so briefly about that spike right there. That's the Paleocene-Eocene thermal maximum. Next, we want to talk about the early Eocene climatic optimum, this broad stretch of very warm time, 2-3 million years, depending upon who you want to ask. The point is it's hot for a long time. Okay? In order to look at this interval, you don't go to the Bighorn Basin. Instead, you go to the Wind River, Green River, and Uinta basins. There is a little bit down here in the Piceance Creek Basin, but it hasn't been well studied yet, so we're just going to kind of ignore it. So we're going to start talking about the Wind River and the Green River basins because that's the better sampled interval. That's where most of the fossils have come from. What we're looking at over here is oxygen data from the ocean again. So these are again 4 AMs. And what we're looking at is the interval of time from about 53 million years to about 49 million years, where the oxygen is furthest to the right, which means it's really warm. So you can see it's a broad warming and then cooling trend. It covers this interval of time, basically. The early Eocene climatic optimum goes from what we would call the WA6, or the Lycidian, so late Wasatchian, which means 42 and a. If you don't study North American land mammal ages, that's a bunch of gibberish. That's fine. The idea is we have a 4 million year interval here where it gets hot, and we've tracked it quite well, going from Wasatchian into Bergerian. Okay? Oh my gosh, that looks fun. Okay, so complex image, but again, nothing we haven't seen. There's dates, there's the Eocene, the subdivisions of the Eocene that are North American land mammal ages, which are further broken down into zones. Great. We're interested in this stuff right here. Okay? And this gives you an idea of what kind of sediments you're going to find depending upon which set of basins you go into. Overly technical, we're not going to worry too much about it. The point being, we have really good stratigraphic control between these basins. We have a really good, even if you cross the mountains and there's no rocks linking up each basin, you can walk across the other side, start finding some animals, and get a really good idea where you are. Okay? So here we are in the Wind River Basin, just north of the town of Lysite. It's a stop sign. It has one store. And so this is the kind of color I'm used to when I go out and I look at fossils. This is what I expect to find. And so this is Richard Stuckey down here from the Denver Museum of Nature and Science. He's surrounded by high school students and a couple of younger kids. And this is part of a high school program where you get students out into the field in the summers, and they work for the museum, and they get exposed to science. And they're involved in everything from finding the fossils to cataloging them. It's a fantastic program, absolutely amazing. And so where we are is just north of the town of Lysite, and we're finding stuff like this. This is what we're going out there to find. We're surface collecting. There's no quarrying going on. It's basically you get down on your hands and knees, or you walk really bent over to the close to the ground, and you go looking for teeth. And the Wind River Basin, various parts of the Green River Basin are absolutely chock full of this stuff. It's easy to find. This is Hyopsodus, a nice little condylarth, very, very common. This is the most common thing you could possibly find out there. But that's typical of what you're going to find, and obviously fitting in the palm of your hand. The teeth are only 1 millimeter or so long, not really that big. Okay? This is the Lost Cabin section. This is the type section for Lost Cabinian or WA7 interval. And this is an absolutely amazing locality. Never mind the colors, which are absolutely fantastic. They go from the brown, the oxidized iron down here to the purples of the manganese. And then they have the intervals of white as well. So you have various paleosols. The really neat thing about this is you can walk down in there, stand on a bench, and you can pick out piles of bone from 100 feet away once your eyes get tuned. It's impossible to go down there and not, it's better than Dinosaur Provincial Park. It's amazing. Okay? This is the only place in the world I've ever gone to find mammals like this. Most of my time is spent looking for 2 or 3 teeth a day, and I think that's a great day. This is cheating. But that's why they have so many specimens. So really fun stuff. Now, what you're actually looking at, for the most part, in terms of paleo environments, is a river-dominated system. So you have lots of nice channel cuts. So this is a nice sandstone layer, a sandstone lens, that's been deposited by a stream, or in this case, probably a river. And it's surrounded by much finer grained mudstones, which are going to be overbank or floodbank deposits. Okay? So that's what you're mostly going to see. So there's our river cut right there if you wanted to look for it, and that's fantastic. Okay? And you can actually see places where these two different kinds of rock interplay. So over here we've got a sandstone. This is a river, this is a channel sandstone. And then we've got these beautiful mudstones out here that are overbank deposits. And what we've actually, these are pickaxes here. We've been trenching through the junction between the two to try and establish what the relationship between the rocks, whether it's being incised or whether they're coeval. But the really neat thing is that you can draw the boundary, and you can see this nice package of sandstone over here. As I'm taking this picture, we're sitting over here, the 5 of us, just having lunch. And when the really neat thing about being a paleontologist and going out and doing this is, while I'm sitting here having lunch, this is what I'm seeing. Because I'm sitting there, I know what fossils are there, I know roughly what the rocks mean, and so I'm picturing myself in a jungle well, it's 45 degrees outside and we haven't seen water in who knows how long. You're sitting in the middle of a desert. This is what I'm seeing. And so it's nice to be able to reconstruct this sort of thing. So during the Eocene, during the early Eocene climatic optimum, this is what Wyoming would have looked like. It's kind of, if you've ever driven through it, it's a little different now, as you can probably tell. Okay. This is just, I threw this in for fun. This is Google Maps of the same site I just showed you. I took my image right over here. I was sitting on a little hill looking at these beds with this rock right here. And what you can see is if you stop and look at this entire patch is BR1A in age, not that it matters overly, but what you can see is this patch of brown up here. That's that channel sandstone. And there's another one. It starts right here. It runs like so. Okay? And all of these whites and reds around it, those are overbank, those are flood deposits, floodplains. And so what you can actually do is you color in the channels, and there's my river flowing that way. That's a small channel, and there's the main meander running around it, mapped from space for 50 million years ago, 55 million years ago. Really neat to be able to do that and say, look, I can see, you want to go visit a river? Let's go sit by the river today. And you go sit on top of this bluff and stare at the sandstone. Okay. One of the other things you find in the Wind River, it's not just mammal teeth. What we have here is a whole bunch of pits dug into the hill. Our high school students are just leaving. They've spent the afternoon digging into these pits, collecting fossil leaves. We have paleoflora as well from the same area. And these leaves are very powerful tools when it comes to climate because there are those who can interpret them based on their leaf area and their thickness and the stomatal density and come up with climate interpretations in terms of what the temperature might have been, how much precipitation might have been there. It's very useful information. The tough part is that you very, very, very rarely find leaves and vertebrates in the same beds. The chemistry required to preserve one is very different from the chemistry required to preserve the other. So it makes things a little difficult there. But there's a lot of data to be had. Okay? So there's a palm frond. This is from the Green River Formation, not actually what we're looking at here. But it gives you an idea that if you were to find that palm frond in, I don't know, Alaska, it would say that the climate is significantly different than what you would expect from today. And just FYI, these palm fronds are found in Alaska during the Eocene. So things are significantly warmer, significantly further north. This is just a pretty picture of flowers sitting in a sandstone. Moving on. Okay, so we put all of this together. We put our teeth and our sediments and our flora together, and we can put together an image like this. So we have our time right here. There's the Eocene, all of our breakdown of the Wasatchian. So our early Eocene climatic optimums right here. There's our oxygen data is telling us that's the PETM, and up here is our big broad EECO. That's fantastic. Mean annual temperature based on leaves, it's a little bit of a mess, but the idea is there's a spike in temperature here according to the flora. Okay? There's one over here according to the flora for the Paleocene-Eocene thermal maximum. Mean annual precipitation, well, it's getting more and more seasonal and drier through the early Eocene climatic optimum according to the plant fossils. And then way out here we have the number of mammal genera. So this is species or generic richness. And you can see there's our spike with all of the incoming taxa from Asia or elsewhere up here in the Paleocene-Eocene thermal maximum, and then it stays nice and high and gets a little, you get a little bit of a peak during the early Eocene climatic optimum before it falls off afterwards. So the richness is relatively stable after the Paleocene-Eocene thermal maximum throughout the EECO. This is the kind of thing you can draw when you have all of this data put together, and it's actually relative. So this is a nice, powerful hypothesis that you can now test. Okay? So that's what I spent a fair amount of time doing. We've looked a little bit at some things you might see here in the Green River and the Wind River Basin. I spent most of my time in the Uinta Basin, and the Uinta Basin's a little bit different in that it covers more time. The exposure in those rocks covers more time than both of these basins together, which may seem really weird because they're bigger, but size doesn't matter. I don't know where we're going to go with that. Okay, so early Eocene climatic optimum during that same interval down in the Uinta Basin. This is a place called Raven Ridge, and it's really pretty. So my field work is not hard on the eyes. What you're actually looking at here, again, you've got these beautiful pink bands. Those are overbank, those are paleosols. They're associated with a fluvial-dominated system. And then sitting right on top of them, this is the Green River Formation. That's a fossil lake. Okay? And so what you're looking at here is through time, as you're going up, the lake is expanding to cover the same area. So it's expanding to fill the basin and push all the river-dominated stuff back out towards the edge. Okay? This big rock in the background, that's called Dinosaur National Monument. That's the only time I'm going to use the word dinosaur today. Okay. Actually, it's not true. There's the town of Dinosaur, Colorado. Okay, now we're done. Okay, so Raven Ridge, we just saw a picture of cliffs up here. And if you turn around and look down the ridge, you're going to see something like this. And I love this picture because it gives you a really good idea of just how much time is set up. There's the layers of history that you can read like a book. Stratigraphy is absolutely fantastic here. The fossils are amazing, if incredibly small. We'll get to that in a little bit. And so each one of these little dots represents a fossil locality of varying age depending upon the color. That doesn't really matter. What we've done here is we've been able to collect upwards of 10,000 specimens and through a large sequence of stratigraphic time, which is quite helpful. Okay, so we can actually put in boundaries, Wasatch and Bergerian boundaries, if we wanted to using the biostrat, which is quite helpful. All right. So an example, there's me digging holes again. An example of what you might see, here's the contact between the Wasatch beds below, and there's the Green River up top. These cliffs are quite impressive when you get right underneath them. Here's an example of part of the base of the Green River Formation, shallow water Green River. These are goniobasis snails that are spending an awful lot of time in the shallow water. This is actually a brilliant marker bed that we've walked for upwards of 15 kilometers, so it gives you a nice stratigraphic anchor. Really useful stuff. Okay, an example of the mudstones up close. Every once in a while, as I dug something like 1,000 different holes, I stop to take a picture of one every once in a while. So here's our nice oxidized manganese nodules, glabules up top. It's really nice mudstones, and you get some nice cross bedding in the sandstones when you get up into the lake sediments, the shallow stuff. Okay. The interesting thing about Raven Ridge as compared to the Wind River and the Green River that we've already seen is that you only have small stuff for the most part. The other basins, you collect everything. It's surface collecting. You can quarry. You find lots of big specimens. You can find lots of small specimens if you're willing to crawl around on the ground. Most of the collecting at Raven Ridge was not done by us, the humans. It's actually done by the ants. This is Pogonomyrmex. That's the genus. I forgot his name. It's the western harvester ant. And what the western harvester ant does is it builds these wonderful nests, and up top you have this beautiful little cone. And they bring little pebbles to glue to the top of the cone so that they can protect the rest of the house. The cool thing about that is a lot of these tiny little mammal teeth are exactly the right size for the ants to go and collect for you. Now this introduces some difficulty in that when I go and find an ant hill like this and I find a tooth on it, that's not necessarily where the tooth came from. Ants range significant distance. They actually range about 20 meters in every direction. Straight down, straight up. If they're on a cliff face, they'll go right straight up or to the side. So you have this 40-meter diameter sphere where your specimen probably came from. That's called time averaging where I come from. It can make life difficult, but actually in the case of these specimens, we have enough sediment that we didn't have any trouble with specimens showing up where they were unexpected. So time averaging wasn't a big deal. So this is my colleague Ben Berger doing what we do. You go and you find the ants, and here's the ant entrance over here. And so what you do is you go sit on the other side of the ant hill, and you make sure your shadow doesn't cover the hole. If the shadow covers the hole, the ants get angry, come out and find out why. And they're miserable little animals when they get on you. Okay, I only included this right here. This is the single largest specimen we collected from Raven Ridge. Again, that's millimeters. Not exactly huge. This is Heptodon. This is a small taper, like a taperoid or a pre-taperoid. I took a picture of this simply because this is it. That's 6, 7 teeth right there. That's as good as it gets. That's what we're working with at Raven Ridge, isolated teeth. If you find 2 teeth in a jaw, you're a superhero. Okay. So some examples of what you're going to see, mostly rodents, honestly. Isolated rodent teeth. This is an exciting specimen because it's not just a tooth, it has jaw attached to it. Very exciting. So again, this is a millimeter bar right here, quite small. We have a whole bunch of different kinds of rodents, and they'll become rather important a little bit later. Carnivorous teeth are everybody's favorite because they're pretty. There's not a whole lot of them there, nor would we expect them to be carnivores. We would expect to be a minority portion of any ecosystem. They're not terribly diverse either, but that's not surprising because there's not a whole lot of diversity within carnivora during the early Eocene. There probably actually is at the biological level. At the taxonomic level, we work with the fossils. It's hard to tell them apart, especially from isolated teeth. Okay? Condylarths, it would not be an Eocene ecosystem without condylarths. So Hyopsodus, this is your typical tooth. Hyopsodus is the single most common genus at Raven Ridge. Lots and lots of specimens. Haplomylus, a little less so. We have some early artiodactyls that are basically diacodexis, lots and lots of diacodexis, and this one buhniferous tooth. That's it. Artiodactyls are nowhere near as diverse as they are now, especially in the early Eocene when they're just getting started. There is a fair number of perissodactyls, and this is actually kind of surprising because these are the ants can't carry these. So all of these perissodactyl teeth are actually collected by us from small channel lags we're lucky enough to find. There's a couple of places that we can actually go and find these things. They're relatively rare, but they're pretty cool when you get them. So they're evidence of, I consider this to be a macro specimen. That's what, 6 millimeters long. That's huge. And then omomyids, primates. Primates are a big component of the Raven Ridge fauna. They're very, very diverse, and they change significantly throughout the section. This is our all-star primate specimen. Shoshonius has all of 3 teeth attached to him. Pretty neat stuff. So with all of this data from the Uinta Basin, we can go and we can compare it to what's going on here at, in the Wind River and the Green River Basin. These numbers are a compilation of basically everywhere in North America. So when we get up to 100 plus genera, that's not 100 plus genera in the Wind River Basin. That's 100 plus genera from all of the taxa from that age that we can find in North America. So it would be unexpected if Raven Ridge had the same amount of species richness. And so when we compare them, that's not surprising. There's Raven Ridge down there, species richness somewhere around, or generic richness somewhere around 30 throughout the early Eocene climatic optimum here. And if you go to Woodburn and them, they've got 90 plus, up to 100 plus genera through each interval. That's fine. It's a relatively flat line. There's no statistical significance to those differences in terms of the pattern. In terms of the numbers, yeah, they're totally different, but you're not comparing apples and apples. Okay? One thing we have to remember is Raven Ridge is just small specimens. This is small and large from everywhere. Okay? So the relatively stable richness, does that mean nothing changed over the boundary? That's kind of the question that, okay, that looks really boring when you see something like that in science. Unless you're hoping for a flat line, that's dull. So the question is, well, is it a result of poor sampling at Raven Ridge? Do we need to go back and get more? And so you can run a quick rarefaction analysis. And you can see that what a rarefaction analysis basically is it's a way of testing your data. Based on your methods of data collection, would you expect to find a whole lot more taxa if you sampled a whole lot more specimens? The closer the line is to asymptote, that is, the closer the line is to horizontal, the better your sampling appears to be. These are all relatively close to horizontal. That's pretty good sampling. So whatever differences in pattern or whatever differences in number we're seeing between this and Woodburn, it's probably not a fault of sampling. You could go and sample a whole bunch more specimens and only get a few more taxa. It doesn't look like we're missing much. So if generic richness of small mammals at Raven Ridge was relatively stable during the, that's fantastic. Nothing really changed. Does that mean nothing happened? Well, actually, a significant number of things happened. The other component of diversity, other than just the number of taxa, is how many of those taxa are there. How is the ecosystem split up amongst those taxa? I'm not expecting you to get a whole bunch of names out of this. What we're looking at here is relatively evenly distributed ecosystems, with the exception up here where Hyopsodus is just king of everything. What that shows us is that there's a lot of different taxa and there's a lot of specimens, or a fair number of specimens from each taxon throughout time. If you were to plug this in, this is very hard to read. This is just kind of an example. You get an idea of the general pattern here. That's about all you get. If you plug it into an obnoxious graph like this, what you can see is here's WA35, here's WA6, WA7. So we're working our way through the early Eocene climatic optimum, and things are changing. This is the big one. This is rodents. And you can see whatever's happening as things warm up and we hit our peak warmth between WA7 and BR1A, whatever is happening, the rodents love it. They think it's fantastic. What is it that they love? That's a little harder to do. a lot of these rodent taxa are literally tooth taxa. We know them from their teeth. We don't know a lot about their body structure. We can't necessarily tell if they're arboreal, living in the trees, or if they're fossorial, digging underground, or if they're just little mice scurrying around in the grass. We can't say a whole lot about how they're using substrate. We're in the process of working up some details of their dentition and working out what they may have been eating. So diet partitioning is another way you could look at this. But whatever's going on, Hyopsodus, microcyopods, they're decreasing. Rodents are loving it. The primates stay relative. The primates are over here in blue, the dark blues. They stay relatively stable. There's a changeover from the little guys and the big guys. They just kind of swap spots in terms of abundance. The flora indicates that things are getting more forested. You're going from a broken canopy to a multi-story canopy. So you're getting more forested. You should be finding more of these guys in terms of abundance. It's not necessarily happening. My guess is if you went up into BR3, you might see a little bit more of them before they crash and burn and primates go away for, well, until we show up again. So there is significant change going on during the early Eocene climatic optimum in the way that the ecosystem is partitioned. Generic richness gives us nothing, but the abundance, the relative abundance, is significantly changed. Okay? In terms of first and last occurrences, again, I'm sorry, this is a little bit of an obnoxious image. What we see here is the first occurrence down here, and then when the bar stops, it's the last occurrence. So we're looking at taxon ranges through stratigraphy. And what we see is it looks like there's a massive origination event right down here. Think, wow, that's a lot of evolution. No, actually what that is that's where our first really good site is. And so we have a couple thousand specimens from a site there, and so that's the first time you find things. That's an artifact of sampling. Once you get up here, you're getting a little better idea of where things are actually starting to occur. There's no major spike in first occurrences or last occurrences, no major extinction events associated with the early Eocene climatic optimum. What it is a gradual change through time with richness staying more or less constant. Okay. So we've looked at those basins. That's great. We can kind of take away a couple of points here that based on the general dump of information I've given to you here. Climatic variability is normal. Climate change is not something that's new to us. I don't think that should shock anybody in the audience. Climatic variability is normal. And obviously it does affect ecosystems. Does it necessarily result in a jump or a mass extinction? No. Does it necessarily result in mass originations, invasions? Not necessarily. It can when bridges are involved. Okay? And not all events that include climate change are going to result in the same reaction. Okay? Paleocene, Eocene thermal maximum, massive invasion from a different continent. The early Eocene climatic optimum, the big thing about the early Eocene climatic optimum is that all of the, we'll go from here. All of the specimens, all of the new taxa appear to evolve from taxa that were still in North America. There's no invaders coming in from Europe. There's nobody coming over from Asia. All of the new taxa you see throughout the Wasatch and into the Bergerian are evolved from something that's already there. They may have immigrated from a basin further south or further north or somewhere else, but it's not like you're getting entire orders coming in from elsewhere. Okay? And the last thing is all of those images at the beginning, we have a really robust stratigraphic framework that allows us to ask very detailed questions about stuff like this. And so it's really exciting to be able to go to the Bighorn Basin or go to the Wind River Basin and have such tight stratigraphic control that you can start parsing out hundreds, tens, thousands of years and getting an idea of how individual species change or evolve through time. And with that, I think I will call it a day and ask if anybody has any questions.